I'm David Hartman, and what I've been doing is using new tools to find seizing neurons in mice. I have no disclosures. I'll start with some acknowledgements. Thanks to the NST. It's been an awesome opportunity, particularly Dr. Porter. Thanks to the residency, the Test Lab, and my family, here to demonstrate that babies in sunglasses are always hilarious. So I'll start by saying something you already know, that temporal lobe epilepsy is a major problem. About 1% of the population is estimated to have epilepsy. About 2/3 of them can be controlled with medications, but that leaves 1/3 of patients with refractory epilepsy. And the majority of patients with refractory epilepsy have temporal lobe epilepsy. Just for reference, this is the temporal lobe. That's a cavernoma in the left temporal lobe from a patient with refractory epilepsy. Sitting on L5 might be such a patient with a particularly putative seizure focus. What we would do to nail down, pin down where to resect or ablate or where the seizures are coming from is have our neurosurgery colleagues put in stereo EEGs along pathways such as this, and then we would record them up in the EMU to try to capture something like this. So from these red dots, these red contacts on the brain there, are seizures emanating from, in this case, the left amygdala. Try to use my pointer here. The left hippocampus, the left entorhinal cortex, and the left parahippocampal gyrus. So this is something we do clinically every other week or so, but some groups are able to zoom in on a particular seizure focus. At the end of the typical stereo EEG electrode are microelectrodes, and they can record individual neuron activity in the human brain. And when people do that, what they see is something like this after a lot of analysis. So you can rank neurons by their activity level. So from 0 to 130 or so, each line is a different neuron in that seizure focus. And you can see what these different neurons do during the course of the seizure. The seizure is here in the red line. That's the onset. And what you see is a bunch of neurons at the top have more black dots, which means they increase their firing rate. But there's also a bunch of neurons in the middle that don't seem to change from their baseline. And there's a bunch of neurons at the bottom that actually seem to decrease their firing. And what we see when watching one seizure to the next is a pretty similar pattern of neuron activity. And what I want you to take away from this is that not all neurons change their firing rate, just a subset. And so from that, with my simplistic design of a seizure focus here with 16 neurons, you can imagine a mosaic. So there's maybe a few neurons in red here that fire during the seizure, and there's several neurons in blue that don't really change. And so you have a mosaic within this seizure focus, and the population of neurons from one to the next seizure are pretty similar. They're not exactly the same, but they're fairly similar from one seizure to the next. However, our current treatments basically ignore this mosaic. So we bathe the entire brain in medications. We resect pieces of tissue that have some healthy and some unhealthy seizing neurons, and we put in RNS or DBS to modulate swaths of neurons, some of which are abnormally firing, some of which are not. And these are all effective, but for 1/3 of patients, they are not completely effective. So they continue to have seizures, which leads me to our unmet need and knowledge gap. So we don't know how to specifically target cells that participate in seizures, like the red cells here. And we don't know how to change their firing pattern specifically. So my project in the Test Lab, we're using a tool called SC-FLARE in mouse models to capture this mosaic. So broadly, the way this works is you have seizures and you have light, and the combination together can turn neurons red or green or whatever flavor you'd like. But the way this roughly works is that you have, let's see, a calcium-sensitive protease, the scissors here, and then you have a light-sensitive love domain. And with calcium and with light, you permit a transcription factor into the nucleus, and it transcribes, in this case, a red fluorescent protein. So at the end of all that, you get a bunch of red dots. So in our model, we have SC-FLARE and the CA1 of the mouse hippocampus, and I put kainate and excitotoxin in the amygdala. And we can have a closed loop system where we detect seizures And when the seizure is detected, shine a light. And those two things together should give us this. So this is a mouse brain. This is the hippocampus. You can see a bunch of red dots that represents labeled cells with SC flare. And there's a few different clusters. There's one here, and one there. And we'll talk more about those clusters later. So SC flare helped find an interesting seizure focus called the fasciola cinereum. What we were seeing is, using this technique, this little part of the hippocampus kept lighting up. And we were like, what is this region? This was mostly before I was there, but what was the region was the question, and it turned out to be a place called the fasciola cinereum. You can confirm that with additional labels. PCP4 is a particular antigen. And again, you see a mosaic of some red and some non-red cells. And what's interesting about this is probably nobody would have predicted seizures were coming from here. And so no one would have put an electrode in that particular region of the hippocampus. So what's cool about flare is areas just light up. You don't have to have an a priori hypothesis as to where the seizures were coming from. So my role was to check if seizures would, or SC flare would also label cells in a different model of epilepsy than was previously used in the lab. They were using CA1 focus, where the kainate was, and I chose to do the amygdala. And I basically found the same thing, which is really reassuring, because it could have been that the excitotoxin was just diffusing into the FC, causing it to have a seizure and label. But by doing it so distant, we see that FC is integrated into the seizure network. It's not just an excitotoxic chemical effect. It's actually integrated into the network and lights up in this seizure model as well. Further work from Ryan, the excellent neurosurgery resident, and Dr. Butch, they took this from the bench and said, let's check if humans have seizures in the FC. And so they actually prospectively put SEEG electrodes in the FC of several patients here and found, indeed, the FC is having seizures in humans. So this was sort of before my time, but this is what the FC would look like on an MRI, sort of tucked behind the thalamus and the lateral ventricle. This is the dentate gyrus up here, and the FC is the tail of the hippocampus. And when they have an SEEG electrode in the FC, you see that it has seizure activity along with the anterior hippocampus in this case. So that was pretty exciting. We have a mouse finding where the FC is seizing thanks to SC flare, and it turns out to also be happening in patients. And you can read more in this article that just came out. So in summary, seizure foci are comprised of some cells that participate in the seizure, i.e. change their firing rates, and other cells that appear to be innocent bystanders. SC flare is a new technique developed by the Ting Lab, a lab here that develops amazing tools that can identify cells that participate in the seizure. And this can illuminate new brain regions that are part of the seizure network, like the FC, the fasciola cinereum. And in the future, I want to use SC flare to study transcriptomic differences between the seizing and the non-seizing cells. That's all I have.
I'm excited to take a little time today to talk to you about developmental neurobiology. I have a longstanding interest in neural development, and as a physician scientist and very soon to be fetal and neonatal neurologist, one of my career goals is to understand mechanisms of neural development, both in typically developing children and in children who have congenital malformations of the nervous system. So the most common congenital malformation of the nervous system are neural tube defects, which affect about 3,000 pregnancies annually in this country. So for those of you who haven't thought about embryology in a while, the neural tube is the embryological precursor to the central nervous system. It forms very early in neural development. And problems with closure of the neural tube have significant clinical manifestations. So if you have a failure of closure of the rostral neural tube anteriorly, that leads to problems like anencephaly or anencephaly, which are universally fatal. And then if you have problems of closure of the caudal neural tube down here, that leads to spina bifida. So it includes meningocele, myelomeningocele, both open defects and closed defects. So as you all know, children with spina bifida have multiple different neurological symptoms, motor symptoms, sensory symptoms. But the symptoms that are consistently rated by parents as having the highest impact on quality of life are actually neurogenic bowel. But despite the prevalence of spina bifida and the morbidity associated with neurogenic bowel, we really don't understand the mechanisms underlying neurogenic bowel. Part of that is because the innervation of the GI tract is incredibly complex. So here I'm showing you a diagram looking at the extrinsic innervation of the GI tract. And so by that, I mean axons that are leaving the central nervous system and going out to the GI tract. So you have neurons that live in the brainstem, the spinal cord, prevertebral ganglia, dorsal root ganglia, all that send their axons to different regions of the GI tract. So this is a little cross section here of the GI tract with the lumen down here. So you can see those extrinsic nerves come in as a little bundle through the mesentery, closely opposed to the blood vessels, and then elaborate out throughout the GI tract. But in addition to those extrinsic nerves, you also have these intrinsic neurons, which reside within the walls of the GI tract as part of either the myenteric plexus or the submucosal plexus. And those neurons form the enteric nervous system. So there's very complicated neural circuitry that's going on here in the GI tract. And so when we think about neurogenic bowel, it becomes a very complicated question, sort of what levels of neural dysregulation are leading to those phenotypes. So the overarching goal of my project is to understand how is enteric neural circuitry affected in models of neural tube defects. So to answer this question, I joined the lab of Dr. Julia Kaltschmidt, who is a developmental neurobiologist and really an expert in enteric neural circuitry, and so very well positioned to mentor me through this project. And we use as our system the mouse, which is a genetically tractable model that allows us to look at both enteric neuron structure and enteric neuron function. So the mouse anatomy is very similar to the human. We have our small intestine and our large intestine. And then we're able to dissect the gut out from a mouse and look at it in isolation. And so here on the bottom, this is a movie showing you a colon from an adult mouse that's living in a little bath. And so you can see we can actually visualize peristalsis and even the expulsion of the colonic contents. And so that allows us to quantitate motility patterns in the colon. In addition, we can also look at the enteric neuron structure. So we dissect out the gut, and then we have a little tube, and we make a longitudinal cut here, and then lay it out like an open book preparation. And that allows us to look at the enteric neurons themselves. And so we can see these neurons reside in ganglia, coalesced into these ganglia, and that these ganglia are arranged in these long stripes that go circumferentially around the gut. And so both the small intestine and the large intestine, we can see these neuronal stripes. Importantly, we can see this not just in the adult, but also during development. So this is work that was done by Lori Dershowitz, who was an MSTP student in the lab, who showed that early in embryonic development at E14.5, these neurons are sort of scattered, kind of not very well organized. And then as time goes on through embryonic development and into postnatal development, you can see they coalesce into these ganglia that then form into these neuronal stripes. And importantly, Lori showed that a similar neuronal structure also exists in the human. So these are samples of human tissue, small intestine on the top and then large intestine on the bottom at different time points throughout development. And similarly, you can see early in development, these neurons are sort of scattered, not very well organized. And then as time goes on, they coalesce into these ganglia that form similar neuronal stripes. So the mouse recapitulates human ENS development, which validates our use of the mouse as a model. So my goal is to visualize both the extrinsic and the intrinsic neural circuitry of the gut during development. So as proof of concept here, I'm showing you that I've been able to visualize both in the same tissue. So these are sections of mouse duodenum with our open book preparation. So early in development at E13.5 on the left and then going across on E16.5. And then to start with, in green, I'm showing you QCD, which is a marker of neurons. So you can see early in development, I hope you can see, I don't know how well the images project, but you can see they're kind of scattered throughout, not very well organized. And then as time goes on, they coalesce, and then by E16.5, you can visualize really nicely these circumferential stripes going across the tissue. So that was sort of a redemonstration of work that Lori had done. And then I was able to show that in addition to those intrinsic neurons, I can also visualize extrinsic innervation. So as an example of that, I'm showing you here the V-glut2-Cre, which is a marker of vagal sensory axons. So these are neurons that reside in the nodose ganglion in the neck, and then have bipolar axons that go up to the brainstem and out to the peripheral organs. And so here we can see those axons entering the peripheral organs in magenta. So at E13.5, they're just sort of barely arriving. And then as time goes on, they innervate in this proximal to distal pattern and elaborate more over time. So you might be able to see that a little bit better with these high resolution images here. So this is E13.5 on the left and E15.5 on the right. So early, you can see those axons are present, but kind of just arriving, very simple, not very branched yet. And then as time goes on, there are a lot more axons present. They're more complicated. They're more branched. That's quantified down here on the graph on the left, just showing you total axon length in a given area of tissue. And then I hope you can also appreciate from this picture, there seems to be a sort of relationship, a patterning between the enteric neurons in green and the axons in magenta. That they're following, they're sort of connected to each other in a way. So that's quantified down here on this graph on the right. I was able to quantify sort of the total length of axon present in the tissue, and then determine what percentage of the axon is connected or opposed to an enteric neuron. So what you can appreciate is that the majority of the vagal axon is actually in contact with an enteric neuron, and that increases during time. So I can look at the extrinsic, I can look at the intrinsic, and then my goal is to bring this forward into models of neural tube defects. So I'm using as my model mutations in the planar cell polarity protein Frizzled 3. So planar cell polarity is a very important signaling pathway in developmental biology. It's important in polarization of epithelial tissues and also important for axon guidance in a variety of different areas of the central nervous system. And you can also see in this picture, it's important for neural tube closure. So this is the control on the left. This is a mouse pup. You can see a nice long tail, little feet here. And then these embryos at E10.5 or so, nice, typical, well-formed. And then in the homozygous mutant, you can see they have this curly tail phenotype, which is very characteristic of mouse models of neural tube defects, indicating some problems with patterning in the caudal end. And then you can see this pretty obvious anterior neural tube defect as well, similar to anencephaly. Interestingly, there's also some evidence to suggest that Frizzled 3 is important in patterning of the enteric nervous system as well. So this is a control mouse on the top and then the Frizzled 3 mutant on the bottom. On the left, you're seeing the whole gut dissected out, so stomach here and kind of going along this way. And on the right, this is zoom in on the tissue, showing you neurofilament staining. So in the wild type, you can see you have these nice, thick, long axon bundles that are going longitudinally down the gut. And on the bottom, you can kind of get the sense those axons are thin and disorganized. So there seems to be some hint that Frizzled 3 is important to organization of at least the axons in the ENS, but we haven't been able to visualize the enteric neurons themselves in these mutants. So this is exceedingly preliminary data, hot off the presses from last week, but so far it does look like Frizzled 3 is important in the organization of the enteric neurons. So this is sections of mouse duodenum at E16.5. You have the wild type on the left, where you can see those nice long stripes kind of going throughout the tissue. And then on the right is the homozygous mutant, and there's just a zoom in here so you can see a little more clearly. So neurons are clearly present. They do seem to be coalescing into ganglia somewhat, but I hope you can appreciate there also seems to be like some cells that are sort of just hanging out by themselves, not present in ganglia. And even when we look at the ganglia, they're sort of disorganized and not really forming these nice linear stripes. So moving forward, I'm planning to take this preliminary data as the basis for my K-12 application. And so my goals are to first look at enteric neuron structure and function in genetic models of neural tube defects using the Frizzled 3 mutant I showed you, and then I have another mutant in the lab as well. And then also look at enteric neuron structure and function in teratogenic models of neural tube defects. So planning to use in utero exposure to valproic acid as my model there. And then down the line, my hope is to build upon this in order to be able to visualize the extrinsic innervation of the GI tract in these models as well.